Whether you're a bioprocess engineer hunting for the perfect scaled-down model, a biotech founder architecting the leap to commercialization, or a scientific analyst who craves raw technical truth, you've reached the technical edge. In this space, we live by a single mantra: the process is the product. Welcome to the Biomanufacturing and Fermentation Technology podcast with Prasad Ernala. Hit subscribe and ring the bell to join a global community and stay at the vanguard of biotech translation. Let's build the bioindustrial future together.
In this Part-3 analysis, optimization, validation, and scale-up with AI of 4 DOE sessions, we confront the hardest question in experimental science. How do we turn data into defensible decisions without being misled by elegant models? Core points from Lesson 3. Data quality sets the ceiling for AI performance. Inconsistent inoculum history, uncontrolled oxygen transfer, sensor drift, or poorly logged deviations will be amplified by models rather than corrected by them. Classical statistics remain the backbone of interpretation. ANOVA, interaction terms, and response surfaces provide the structural discipline that keeps AI-generated insights biologically meaningful. Not every outlier is an error. Some deviations represent real metabolic state shifts or transport limitations. The decision to exclude or reinterpret data must be grounded in process understanding. Optimal conditions are often fragile. Robust regions with flat performance plateaus are more valuable than sharp peaks that collapse under small perturbations or scale-up stress. Validation requires falsification, not confirmation bias. Independent runs, reproducibility checks, and sensitivity testing determine whether improvements are real or model artifacts. Scale-up physics must be embedded early. Mixing time, oxygen gradients, substrate distribution, and co-stripping redefine performance at volume. Bench-scale optima do not automatically survive production reality. AI is most powerful in scenario testing, robustness simulation, and workflow automation. It becomes dangerous when used for unconstrained extrapolation beyond biological and engineering limits.
Welcome to the debate. Today, we are stepping right into the pressure cooker of the biotech industry. We're talking about the incredibly difficult, high-stakes process of bioprocess development, turning messy, chaotic biological data into defensible, multi-million-dollar manufacturing decisions. And right now, that world is trying to figure out if its newest tool is a savior or, well, a hallucination.
It's a question that usually costs about $50 million to answer incorrectly.
Exactly. The central question we're wrestling with today is this: how do we integrate artificial intelligence into bioprocess development without fooling ourselves? We're looking at the tension between the promise of AI-accelerated optimization and the stubborn physical realities of fermentation, data quality, and scale-up physics. I'm the advocate, and I'm arguing that AI is the only tool powerful enough to handle the complexity of modern biology.
And I'm the dissenter. And my argument is simple. Biology is noisy, and AI loves to amplify noise. I think we're in danger of handing the keys of a very expensive car to a driver who has never actually seen the road.
I see why you'd say that given the hype cycle we're in, but let's look at the alternative. For decades, bioprocess development has been driven by intuition, by luck, really. You run a shake flask, you check the final yield, the titer, and you guess what to change next. But today, we aren't just looking at endpoint titers. We have PAT.
Process analytical technology?
Right, essentially eyes inside the reactor. We have Raman spectroscopy probes, capacitance sensors, and off-gas analyzers streaming high-density data every single second. The sheer volume of multivariate interactions just exceeds human intuition. We are drowning in data, and without AI, we're just skimming the surface.
The volume of data has increased, sure, but does more data mean better decisions? I look at these AI models applied to bench-scale data, and I don't see clarity. I see prediction fantasies.
That's a strong phrase. How so?
Because of the garbage in problem. Let's start at the bench, where the data is actually created. In a perfect world, every experiment is executed flawlessly. In the real world, you have a technician who creates a slightly different inoculum density or a shaker table that stops for 10 minutes because someone, bumped the plug.
Variations happen, yes.
But here's the problem. If those deviations aren't logged perfectly, and let's be honest, they rarely are, the AI doesn't see error, it sees discovery. It looks at that dataset and learns that a 10-minute agitation pause improves yield. It overfits the model to the noise. It learns the artifacts of a bad experiment and presents them to you as biological insight.
That is a risk, but you're describing a data hygiene problem, not a technology problem. In fact, I'd argue that AI is actually our best tool for cleaning that mess up. We're seeing incredible results with soft sensors.
Explain that for the listeners who haven't used them.
A soft sensor is basically using software to measure something hardware can't. We use multivariate analysis to correlate messy, noisy signals, like off-gas and pH trends, to predict invisible attributes like glucose levels or product concentration in real time. It finds the signal in the noise, and more importantly, it handles the gaps.
Gaps?
Missing data. Instruments go down. Probes fail. Research shows that for dynamic series, modern algorithms can impute, mathematically fill in, up to 30% of missing data entries without breaking the model's predictive power.
Wait, pause there. Impute up to 30%. You are saying that if I lose a third of my data because a probe failed, the computer just invents it?
It's not invention, it's statistical reconstruction based on the covariance structure of the remaining data. It knows how the variables move together, so it can fill in the blanks with a very high probability.
To me, that sounds terrifying. You are patching holes in the map with what you think the terrain looks like. And this leads to my biggest issue with this whole data cleaning mindset, the failed run trap.
Which is?
In the real world, runs fail. Contamination, controller drift, pump failures. A human looks at that and says, trash it. But an AI, especially an anomaly detector, might look at a sudden drop in growth and flag it as an outlier to be removed. But what if that wasn't a pump failure? What if that was the biology hitting a stress threshold?
Like overflow metabolism?
Exactly. The cells switch metabolic states because they're stressed. The growth rate drops. If you have an AI cleaning your data, it might say, hey, this point deviates from the expected curve. Let's delete it to improve the R-squared.
But that is exactly where the partnership works. The AI flags the anomaly faster than a human scanning spreadsheets. It forces the scientist to look at that timestamp. It says, look here. Then the human expert cross-references it with the execution record. Was there a feed pump issue? If yes, it's an artifact. If not, then it's a biological state we need to investigate.
Ideally, yes. But the temptation is to smooth the curve, to make the model fit. When you delete that outlier, you might be deleting the most important discovery, the cliff edge where the process dies. You end up with a model that predicts a smooth, happy path that just doesn't exist in reality.
You're talking about robustness now. And I agree we shouldn't be chasing fragile peaks. But this is where I think AI beats classical statistics hands down. We need to move from the bench to the computer to the modeling itself.
Oh, that's blasphemy. I will take a classic ANOVA table any day.
Hear me out. Classical statistics, response surface methodology, looking at contour plots, is great for 3 variables: temperature, pH, dissolved oxygen. But biology has 20. We have amino acid feed rates, induction times, pressure, agitation strategies. The dimensionality is massive. There was a recent case using large language models, specifically ChatGPT-4, to orchestrate experimental designs for microalgal growth.
Orchestration is fine, but what about the math?
The AI didn't just organize the spreadsheet. It successfully identified significant linear and quadratic terms. It found complex, nonlinear curvature in the design space that a human, using a standard quadratic model, would have missed completely. It accelerated the path to a high predictive R-squared.
But can you explain why it picked those points? That's the problem with the black box. With a classical ANOVA analysis of variance, I can see the p-values. I can see the temperature interacts with pH with 95% confidence. It's interpretable. When you start stacking neural networks, you get an answer, but you lose the why.
I think the black box reputation is becoming a little outdated. We have feature importance maps now. We can see which variables are driving the prediction. And more importantly, we can use desirability functions to balance trade-offs in a way human brains really struggle with.
Desirability functions are dangerous. I've seen them ruin projects.
They're standard practice. You weight your goals, say 60% importance on titer, 40% on purity, and the math finds the sweet spot that satisfies both.
But that one scalar number hides the trade-off. It compresses all that complexity into a single score. You tweak those weights just slightly. Say you value titer just a little bit more, and the calculated optimum just jumps across the map.
But that's why you do sensitivity analysis, though.
But the AI presents it as the answer. It says, here is your optimal condition. It offers an illusion of optimality. I would much rather look at the raw contour plots myself. I want to see the shape of the mountain. I want to find a plateau, a broad, flat region where I can be a little sloppy with the controls and still get a good product.
That is exactly what AI allows you to do, though. We can run thousands of in silico simulations. We take the model, add virtual noise to the inputs, simulate a pump that's 5% off, or a temperature probe that drifts, and see if the process crashes.
So you stress test the model?
Exactly. We can mathematically identify those robust plateaus in a way that just eyeing a chart never could. We can find the shoulders of performance where the risk is low, rather than the peak where the risk is high.
If the model knows where the cliff is. And that brings us to the ultimate reality check. Scale up.
The move to the big tanks.
The 5,000-liter reality check. This is where I think the AI hype train derails. You train these models on bench-scale reactors, small, perfectly mixed, highly controlled. Then you move to a production tank. Do you know the mixing time in a 5,000-liter mammalian cell vessel?
Depending on the geometry, it can be anywhere from, 80 to 180 seconds?
Right? 3 minutes. In a bench reactor, mixing is instantaneous. In a big tank, a cell can spend 3 minutes suffocating in a low oxygen zone or getting blasted by high pH near the feed port before it circulates back to safety.
The gradients.
Exactly. Massive gradients of pH, dissolved oxygen, and substrate. These physical stresses do not exist in your training data.
That's true. Bench data is clean, plant data is dirty physics.
So your AI has never seen the weather it's about to fly into. It predicts a yield based on sunny day bench data and then fails catastrophically in the hurricane of the production tank. It is blind to the physics of large tanks. You're asking it to extrapolate, and we both know data-driven models are terrible at extrapolation.
I concede that point. If you just feed it distinct data points, you're right, it's a fantasy. But that is why we don't use naked AI anymore. We use hybrid models.
Hybrid meaning?
Meaning we embed the physics into the code. We take the known engineering equations, mass transfer coefficients like K to A, mixing time correlations, Reynolds numbers, and we use those as constraints in the neural network.
So you're teaching the AI the laws of physics?
We're forcing it to obey them. The AI solves for the biology, the stuff we don't fully understand, but the engineering math constrains the environment. We can ask, if the mixing time shifts to 100 seconds, what happens to the growth rate? The AI interpolates the biology within the constraints of the physics we understand.
So you're using physics to keep the AI on a leash?
Exactly. It prevents the AI from proposing physically impossible scenarios. It turns it from a prediction engine into a risk assessment engine. It bridges the gap between the flask and the tank.
I can respect that approach. It sounds much more grounded. But it still requires that you validate the prediction. And this is where the human element fails. We get a fancy chart from the model, it looks sciencey, and we treat it as truth.
Which brings us to the workflow. How do we actually use these things?
For me, the most valuable tool isn't the model itself. It's the falsifiable hypothesis.
I agree. Every model prediction is just a hypothesis waiting to be tested.
Exactly. If the confirmation run doesn't match the AI prediction, the model is wrong. Period. You don't tweak the data to fit the model. You refine the model structure or identify missing factors.
And that validation step is critical. We need confirmation runs at the predictive optima, and we need to be rigorous about them. Independent inoculum, independent media batches. But the goal of the AI here is to make that process repeatable. Imagine a library of reusable prompts and models.
A library of prompts?
Yes. Instead of every scientist reinventing the wheel, building their own shaky Excel sheet, we have standardized prompts for analyze this ANOVA table or design a central composite DOE. This standardizes the analysis. It ensures that everyone is using the same rigorous statistical criteria.
I see the efficiency there, but I worry that standardizing the analysis standardizes the lack of critical thinking.
How so?
If the routine is automated, people stop checking the work. If the AI says significant interaction between feed and temperature, do you check the residuals? Do you look at the raw data to see if it makes biological sense? Or do you just copy and paste the p-value into the report?
That is a valid concern. It's the GPS effect. You follow the line on the screen and drive into a lake.
Exactly.
But that is a training issue, not a technology issue. We need to train our scientists to translate those statistical outputs into biological language, to explain that a negative interaction between temperature and feed rate isn't just a number, it's oxygen limitation. The AI highlights the pattern, the human provides the mechanism.
That translation is the hardest part, and AI systems often hallucinate those explanations if they aren't grounded in a curated knowledge base.
Which is why the continuous improvement aspect is so vital. As we gather data across projects, historical DOE results, scale-up outcomes, we can feed that back into the system. We can teach the AI that host system A is oxygen sensitive, so it stops suggesting high feed strategies that will crash the culture.
I'll grant you that. If we can encode the scars of past failures into the model so we don't repeat them, that's valuable. But we have to be honest that we're building a partnership, not a replacement.
I think we are converging on a synthesis here.
Perhaps. I still maintain that AI amplifies the consequences of poor judgment. The optimum is a moving target.
And I still maintain that without AI, we're flying blind in a data-rich world. But you're right. The true value lies in using these tools to find a robust operating window, not a single magic number.
We must value reproducibility and control over marginal peak performance. If the AI helps us find a boring, stable, reproducible process, then I'm on board. But if it's chasing the highest yield at the cost of stability, it's a liability.
I think that's a fair characterization. The future of bioprocess development isn't AI alone. It's an explicit partnership. Human judgment plus classical statistics plus constrained AI.
Constrained is the key word.
We've covered a lot of ground today, from the dangers of overfitting and the trap of failed runs, to the battle between classical ANOVA and AI-assisted optimization.
In the cold, hard reality of scale-up physics that no algorithm can wish away.
Indeed. It's clear that while AI offers tremendous acceleration, it demands a higher standard of experimental hygiene and mechanistic understanding, not a lower one.
Precisely. The tool is only as good as the hand that wields it.
A fitting place to leave it. Thank you for listening to The Debate. We hope this exchange has illuminated the complexities of integrating AI into your workflows.
Keep your eyes on the data and don't let the model fool you.
Until next time.
Hi there, thank you for staying with me until the very end. If these technical insights help bridge the gap in your own process, please hit subscribe and make sure to hit the notification bell. Stay connected for our next deep dive into the realities of biomanufacturing. I am Prasad Ernala. Thanks for listening.